Then there is the danger that the ignorant man may easily underdose himself and, by exposing his microbes to non-lethal qualities of the drug, make them resistant. Hi, I'm Elise/AquilaCat here, and welcome to the second video in my Elise's Elucidations series. In that introduction quote, Alexander Fleming was referring to penicillin and describing the process of antimicrobial resistance. Antimicrobial resistance is when microbes like bacteria, fungi, protists, and viruses no longer respond to medicines that used to work effectively against them. They become harder to kill, and their infections harder to treat. Since the discovery of penicillin, countless lives have been saved through being treated with antibiotics. For a while, scientists were able to discover antibiotics through soil samples, a method utilized throughout the 1940s through the 1960s. The issue is that after a while, no new compounds were able to be found with the technology they had available at the time. And with the use of antibiotics naturally came antibiotic resistance. So penicillin was used during World War II to help soldiers fight infections, but by the 1950s, bacteria were already becoming resistant to it. And then in the 1960s, we got Staphylococcus aureus, which causes staph infections, becoming resistant to methicillin. And this is when MRSA first started, and that stands for methicillin-resistant Staphylococcus aureus. In recent decades, unfortunately, more antibiotics have become less effective as microbes adapt to the medications, reducing the medicine's effectiveness in treating an infection. This happens in a few different ways. So some bacteria have genes that are naturally resistant to antibiotics, and this is called innate resistance. Other bacteria develop resistance over time as the microbes evolve, and yet others gain resistance from viruses that infect bacteria, called bacteriophages, through a process called transduction, which is when the bacteriophages transfer resistant genes to the bacteria that they infect. Now, agencies around the world started considering antimicrobial resistance to be a serious threat around the early 2000s. The World Health Organization reports that in 2019, antimicrobial resistance was directly responsible for over 1 million deaths worldwide and more than 35,000 deaths in the USA alone every year. In the USA, it's estimated to cost $1 trillion in additional healthcare costs by 2050 and $1 to $3 trillion in GDP losses by 2030. Now, there are many different people in many different fields all working together to try and solve this global issue. Now, discovering new antibiotics that are effective against these resistant microbes is just one of the ways to combat antimicrobial resistance, and it's the one that I'll be focusing on today. Now, before, I mentioned that new antibiotics were no longer being discovered in soil samples after a while, but the focus of this video, the iChip device, has been incredibly helpful with discovering new antibiotics in soil and water samples that until fairly recently, these microbes couldn't be cultured in a lab and studied before through traditional culturing methods such as using petri dishes and incubators. So iChip is short for isolation chip, and it's the first diffusion microchamber array, known as DMA device, that aids in culturing bacteria, and it was developed by Novobiotic Pharmaceuticals. The Great Plate Count anomaly is an issue with laboratory work where many species of bacteria, 99% of them in fact, can't be properly cultured in the lab on a petri dish or in an incubator because it doesn't meet the right growing conditions for the bacteria. And if they can't be grown in a lab, then they can't be studied, and any benefits they might have go undiscovered. The iChip device helps with this problem by culturing the bacteria species while they're still in their natural environment, and this is known as in situ cultivation. So the process starts with diluting the soil sample, or in the case of water samples, vortexing or lightly sonicating the water, and then suspending it in liquid agar, resulting in a single cell in each of the iChip's compartments that are called wells. This is where the isolation part of the name comes from. Once the agar has solidified, then the plastic plate is then enclosed in polycarbonate membranes and put back into the environment the samples were taken from, and then it's left there for anywhere from 1 to 4 weeks. The semi-permeable membrane allows some things to go through and others not, so it allows the nutrients within the natural soil to enter into the iChip, allowing the bacteria in the wells the nutrients that they need to thrive, while preventing the bacterial cells in the wells from leaving the iChip device. So the process ends when it's finished. The cultivated samples are then extracted from the iChip, usually with the use of like a sterile toothpick and usually under a dissecting microscope. And then the microbes are identified through subculturing or gene sequencing. As of 2023, Novobiotic Pharmaceuticals has collected over 64,000 bacterial isolates that couldn't be cultured prior to the development of the iChip, and by researching those, several promising antibiotics have now been found. So this base design of the iChip device has been improved over time to suit the needs of various research teams. For example, a modified iChip called iChip, FACS, and this stands for fluorescence activated cell sorting. This one optimized the efficiency of the original iChip design, since the original design had the limitation of each of the wells not being guaranteed to have a single cell per well. Some of the wells could potentially have no cells at all, and others might have multiple cells. And there are also modified versions of it referred to as iPlates that are affordable and customizable enough for students and teachers to utilize. Now, let's touch on some of the specific antibiotics that have been discovered thanks to the use of the iChip device. So, first and foremost is Teixobactin. In 2015, the iChip device was used with soil samples to discover Teixobactin, which is produced by the proteobacteria Eleftheria terrae, related to aquabacteria, which itself was also discovered through the iChip technology. Teixobactin works by attacking membranes containing lipid 2 and lipid 3. This prevents the cell wall from developing and growing and leading to structural weakening. It also thins out the cytoplasmic membrane. And since the cell wall is what gives a cell its shape and protects it, without the cell wall or with a severely weakened cell wall, the rest of the cell's contents would spill out and the cell would die. So antibiotics that attack a bacteria's cell walls are effective at killing the bacteria. One of the interesting things about Teixobactin is that it was the very first antibiotic discovered through the use of the iChip device. And the lipids it bonds to are not present in eukaryotes, a group that humans are in, so it avoids the side effects that other membrane-attacking antibiotics have. Now, Teixobactin is effective against gram-positive bacteria and Mycobacterium tuberculosis, which is a huge global concern right now, which you might be aware of if you follow John Green's work and videos. And by the way, if you haven't seen his video on tuberculosis yet on the Crash Course channel, I highly recommend it. Current antibiotics in use with similar effects as Teixobactin are oxacillin and vancomycin. And vancomycin was effective against Staphylococcus aureus once it started to become resistant to methicillin, when it became MRSA. These are the antibiotics that Teixobactin would replace. And it's also shown to be effective against Streptococcus pneumoniae and vancomycin-resistant enterococci. It's also proved effective against Clostridium difficile, so C. diff, and Bacillus anthracis. Inhalation anthrax is what that one causes. In 2020, a study using rabbits tested Teixobactin's effectiveness against inhalation anthrax, and the infected rabbits that were treated with Teixobactin and levofloxacin all survived, which supported its potential eventual use to treat humans infected with inhalation anthrax as well. Although Teixobactin resistance is possible with bacteria in the same environmental niche as Eleftheria terrae, Teixobactin resistance isn't likely to happen when used in human medicine. There is still more research to be done on it before it can be approved for human use against antibiotic-resistant bacteria, but the results are promising for its eventual clinical use. It's been almost a decade since its discovery, and despite many promising studies, it's still in the process to be approved. This is partly due to technical issues, like how labor-intensive it is to synthesize Teixobactin. Other reasons for Teixobactin taking so long to be approved is because it's expensive at $4,000 per dose, and it also needs to be able to compete against other effective antibiotics on the market to justify this expense. And as you can see from this example, this is why the lull in new antibiotics being researched and discovered has been so detrimental while dealing with the general stress of trying to beat the clock to find new ways to treat resistant bacteria before the resistance becomes too widespread and untreatable. The next one up we'll talk about is Clovibactin. So Clovibactin was discovered in soil samples from North Carolina and is produced by the gram-negative Protobacterium Eleftheria terrae subspecies Carolina. Note the similarity to the bacteria that produced Teixobactin. Clovibactin attacks bacterial cells by wrapping around the cells to neutralize them, And it does this by binding to pyrophosphates in lipid 2, lipid 3, and molecule C55pp. All of these molecules contribute to the construction or the synthesis of bacterial cell walls. Now, just like with Teixobactin, clovibactin is effective against gram-positive bacteria, like those that cause tuberculosis and MRSA. In a study treating mice with clovibactin, it was effective against Staphylococcus aureus and against Enterococcus faecalis and Enterococcus faecium. Also note that bacteria that produce antibacterial compounds tend to have compounds that attack bacteria unlike themselves, like how Eleftheria terrae is a gram-negative bacteria, and the antibiotics produced from it attack gram-positive bacteria. And really quick, while we're on this topic, bacteria are classified as gram-positive versus gram-negative based on how well they retain dye that's used in gram staining. So gram-positive species stain a violet color, whereas gram-negative species stain a red or pink color. They also have different characteristics relating to antimicrobial resistance, like how gram-negative bacteria have a double membrane surrounding the cell and have an active efflux system, both defenses of which make it more resistant to antibiotics than gram-positive bacteria. This is why finding antibiotics that are effective against gram-negative bacteria is especially important with battling antimicrobial resistance. And so back to clovibactin. Bacteria becoming resistant to clovibactin would be difficult for them to do, and it has few side effects since its mode of action, or way of attacking, is so targeted to bacterial cells and not to human cells. However, more testing needs to be done, as this is a more recent find through the iChip device. And then there's lasomycin. Now, lasomycin is produced by the bacteria Lentze kentuckiensis. This antibiotic has a narrower scope than the others discussed so far, as it's really only effective against mycobacteria. And it kills these bacteria by targeting and binding to the enzyme CLPC1, which is essential for microbacteria survival, but not for the survival of other types of bacteria. And then instead of damaging this enzyme, lasomycin actually activates it to proceed with ATP hydrolysis, which is where water is added to one molecule in order to break it up into smaller molecules. And from what I understand, it seems like CLPC1 combined with ATP hydrolysis controls the selective killing of proteins in mycobacteria, and by having lasomycin activate it while simultaneously also interfering with ATP hydrolysis, it stops selectively killing proteins and instead goes into overdrive, just killing proteins with reckless abandon, which causes the bacteria to die. And I'm not gonna lie, my eyes started glazing over a bit while trying to make sense of this in the research, so I might be wrong with this explanation, but I think that's what's happening here. And then there's hypeptin. It's produced by Lysobacter species. It's similar in structure to Teixobactin and effective against gram-positive bacteria, including Staphylococcus aureus, with decent effectiveness against mycobacteria and vancomycin-resistant enterococci, also known as VRE. And then the way it attacks gram-positive bacteria is by blocking cell wall biosynthesis, so the construction and growth of the cell wall, making the cell wall weak. And then, as with all of the ones mentioned before, more studies do need to be done. So the discovery of antibiotics isn't the only thing that the iChip device has been helping with. It's also useful even for just cataloging the diversity of microbes in water and soil samples in general, which is the first step to digging deeper into the mysteries that they might hold. So a 2022 study obtaining samples from an estuary in Portugal noted that detecting two specific genes in their samples, PKS1 and NRPS genes specifically, was an important step to determine a microbe's potential use in biotechnology, and that the iChip allowed them to narrow down their focus onto only microbes containing those genes. This same research team has gathered samples in other areas of Portugal and published more papers on the results where they screened their microbial findings for antibacterial and antifungal effects. Publications like this appear to provide great foundational information for other researchers to springboard off of and narrow down the scope of their own research into any of the specific microbes that were found. New species in general are also being found with the iChip, such as Hanstruopera marina and Hanstruopera flava, which were found on Maishan Island in the East China Sea and were introduced in a 2022 paper. They used a modified iChip for this. This entire genus, part of the family Flavobacteriaceae, was just discovered and introduced in 2015. A paper from 2019 detailed how the iChip device was used to isolate bacteria obtained from seaweed samples from the Irish coast as they decayed. Enzyme extraction is difficult from seaweed because the cell wall is so difficult to break down, and the enzymes obtained through the use of the iChip showed more efficiency than commercial options in degrading the cell wall so that the metabolites we want from the seaweed can be obtained for use in areas like agriculture, cosmetics, and pharmaceuticals. And a paper in 2019 detailed using the iChip to find a potentially new species of the genus Alteromonas from giant barrel sponge samples collected in the Bahamas. And it's even being used to isolate microbes from rocks. So a paper in 2019 detailed using a modified iChip to isolate bacteria from brine samples obtained from Triassic rock salt deposits in Northern Ireland. The use of PFA replaced the use of soil for the iChip's culturing environment so that they were able to get it to work. Noteworthy finds here were a strain of Halubrum saccharoverum that had some microbial activity against Pseudomonas aeruginosa, and also just their ongoing study of their findings to determine if any of the isolated microbes are the result of new antimicrobial compounds. And then a paper in 2022 used the iChip on mangrove soil samples from China and found a new strain of Hahella chechuensis. Now, all of these types of studies have just been since 2019, and with how affordable and easy it is to replicate and modify an iChip device in the lab, really any researcher who wants to utilize it in their research can. So it's really cool. And obviously it's been very helpful so far, and it kind of feels like we've only seen the beginning of the discoveries that we can make from using this device. And then wrapping back around to antimicrobial resistance again, ultimately the discovery of new effective antibiotics is only a temporary solution, and a very slow-going one at that. Of all the recent antibiotics discovered and in development in recent years, only cefiderocol and SPR206 have both gained FDA approval and are also effective against multi-drug resistant bacteria that the World Health Organization is concerned about. Microbes will just keep on evolving to be resistant over time, resulting in this endless cycle of antimicrobial resistance, new medicines being found to combat them, and the microbes evolving again to resist the effects and continue to cause problems. The important thing is that we need to keep on looking for solutions for different aspects of the issue of antimicrobial resistance. But, like with all of the new antibiotics being discovered through the iChip device in the last decade, who knows what new benefits we can find out and utilize from these discoveries over time. Let me know if you'd like to know more about antimicrobial resistance in more detail, what else is being done to combat it worldwide, and more about what influences it. Or let me know if there's any other topic you would be interested in hearing recent research about. I hope you are all doing well, and if any of the information in this video piqued your interest, do take a look at the resources linked at the end here and in the description to delve more into the topic. But thank you so much for watching, and until next time, bye.